Hello everyone, and welcome to Vet NeuroJar, the podcast where we explore the fascinating world of veterinary neurology. Today, we're diving deep into a recent study that's shedding light on canine idiopathic epilepsy, or IE, a condition that, much like in humans, can be challenging to manage. This research isn't just about our furry friends, it also has implications for understanding similar conditions in people. This study, conducted by a team of researchers from Ghent University and Nestlé Purina Petcare Europe, looked at the metabolic profiles of dogs with IE, comparing them with healthy dogs. The goal was to uncover the metabolic pathways that might be at the root of this neurological condition. Let's begin with the basics. Idiopathic epilepsy is the most common chronic neurological disease in dogs, and it's a natural model for human epilepsy types with both genetic and unknown causes. This makes studying canine epilepsy incredibly valuable. Both humans and dogs share similar clinical traits, such as the occurrence of status epilepticus, which are prolonged seizures and behavioral issues that can accompany the condition. What's interesting is that the prevalence of epilepsy is quite similar in both dogs and humans. Now, what made this study unique? Well, the researchers used metabolomics, which is a powerful technology that allows us to characterize an individual's biological makeup. It looks at how various factors like disease, the microbiome, environmental influences, and even diet interact and how they affect the metabolism. By analyzing the metabolome, scientists can gain insights into the different biological processes that might be happening in the body and how they might relate to conditions like epilepsy. One of the important advantages of studying epilepsy in dogs is that their dietary environment can be easily standardized. All the dogs in this study were fed the same commercial food, ensuring a consistent dietary background. This is critical because it helps to exclude nutritional factors from potentially skewing the results. That level of dietary control is very challenging to accomplish in human studies. The researchers divided the dogs into three groups: healthy dogs, those with a mild form of epilepsy, and those with a drug-resistant form of epilepsy. The dogs with IE had been diagnosed following guidelines from the International Veterinary Epilepsy Task Force, using different criteria. All the dogs were fed the same adult maintenance diet for at least 20 days before blood samples were taken. Blood samples were then analyzed to measure targeted metabolites and untargeted features using advanced liquid chromatography and mass spectrometry. This is important because it provides a very detailed picture of the metabolic landscape in these dogs. The data was analyzed using various statistical methods to identify significant differences in metabolite levels between the groups. What did the results reveal? The researchers found that the plasma, or the liquid portion of the blood, of dogs with both mild and drug-resistant epilepsy showed distinct metabolic profiles compared to healthy dogs. These differences weren't random. They were linked to specific metabolic pathways. The major pathways that were impacted involved oxidative stress, inflammation, and amino acid metabolism. Additionally, they found that dogs with epilepsy had significantly lower levels of vitamin B6 in their plasma compared to healthy dogs. Let's delve into these altered pathways a bit. First, oxidative stress. The researchers found increased levels of gluconic acid in the plasma of dogs with IE and xanthorenic acid in drug-resistant cases. Gluconic acid is considered a marker for oxidative stress, with studies linking it to brain injury and similar conditions in humans. Xanthorenic acid, also an indicator of oxidative stress and inflammation, showed increases that can be tied to vitamin B6 deficiencies. These findings suggest that dogs with IE experience increased oxidative stress, similar to what has been observed in human epilepsy studies. Additionally, two compounds called carnosine and 2,6-dihydroxybenzoic acid were found to be decreased in drug-resistant dogs. These compounds can protect against oxidative stress and are related to gut bacteria, which indicates that there are a number of factors working together. Now let's consider inflammation. The study showed that inflammation pathways were also significantly altered in dogs with epilepsy. This inflammation can contribute to seizures and might play a role in the progression of the disease. One key finding in this study is that there was a trend toward perturbed pathways related to vitamin B6. The active form of vitamin B6 called pyridoxal 5-phosphate, or PLP, was found to be significantly lower in dogs with epilepsy, both the mild and the drug-resistant types. This is really important, as vitamin B6 is a crucial cofactor in many metabolic reactions, and its deficiency has been linked to epilepsy in children. It's noteworthy that even though the diet was standardized and exceeded recommended vitamin B6 levels, the dogs with epilepsy still showed reduced plasma concentrations, suggesting an increased utilization rather than a simple nutritional deficiency. Let's now shift to amino acid metabolism. The research revealed that several amino acids and their derivatives were significantly altered in the plasma of dogs with epilepsy. Many amino acids were actually found to be increased in the plasma of dogs with epilepsy. Some of these were elevated in both mild and drug-resistant cases, while others were specific to the drug-resistant group. These alterations suggest potential disruptions in the way the body metabolizes amino acids. This in turn has a lot of implications for liver and brain function. Furthermore, 2 glucose-derived acids were also increased in the plasma of dogs with IE. The study also looked at the ratio of branched-chain amino acids, or BCAA, to aromatic amino acids. There were no significant differences in these ratios between healthy and epileptic dogs. However, when the researchers looked at the ratio of kynurenine to tryptophan, they saw a significant decrease in dogs with drug-resistant epilepsy, which points to some interesting changes in tryptophan metabolism. Tryptophan is important for serotonin production, which impacts mood and neurological processes. The alteration in tryptophan metabolism might be linked to the inflammation and oxidative stress observed in these dogs. Another interesting result is that a compound called ethyl-3-indole acetic acid, which is a tryptophan metabolite normally produced by intestinal bacteria, was found to be decreased in the plasma of dogs with epilepsy. This may point to some impact on the gut microbiome or alterations in how the body handles this compound. Additionally, 3 methylated amino acids were also altered in drug-resistant dogs. These amino acids act on the NMDA receptor, which is very important for the functioning of the brain. Now, let's talk about some specific compounds that stood out. 4-guanidinobutanoic acid, or 4-GBA, was found to be significantly higher in the plasma of drug-resistant dogs. This compound can interfere with brain energy metabolism and can be convulsive. It might serve as a marker for how well a dog is responding to anti-seizure medication and is an interesting avenue for further research. The study also showed that N-acetylated amino acids were increased in dogs with IE. These are formed during a process of acetylation, which alters the properties of the molecules. It may be due to an alteration in the way the body is processing xenobiotic compounds, like anti-seizure medications. The researchers may note that the study has some limitations. One is the potential impact of the medications on the results. Also, while the diet was standardized, the actual food intake wasn't precisely monitored, and body condition score of dogs in the drug-resistant group was significantly higher compared to the other groups. Another limitation was that the time between the last seizure and sample collection varied considerably among dogs. The use of owner diaries to record seizures also has its limitations, such as possible missed seizure events. Therefore, future studies should account for those variables by looking at samples from both acute and chronic phases of seizures. So, what does all of this mean? Well, this study has demonstrated that dogs with idiopathic epilepsy have a unique metabolic profile compared to healthy dogs. These changes involve pathways related to oxidative stress, inflammation, and amino acid metabolism. It also revealed lower vitamin B6 levels, which, importantly, had not been previously identified in dogs with IE. These findings provide strong evidence that idiopathic epilepsy in dogs involves significant metabolic changes and is more than just a simple neurological disorder. The information from this study can be used to increase our understanding of the dog as a natural animal model for human epilepsy. It also has implications for further investigation into the potential causal role of vitamin B6 deficiency in epilepsy, and how different pathways can lead to new therapeutic targets. This study also highlights that there are potentially important features that were not yet identified in the database, meaning that there is even more work to be done. Ultimately, the potential implications of this study are huge. By understanding the metabolic pathways involved in canine epilepsy, we can develop better treatments for our canine companions. These discoveries can also be used to inform research into human epilepsy. This makes the study of canine epilepsy invaluable. That's all for this episode of Vet NeuroJar. We hope you enjoyed this deep dive into the complex world of canine epilepsy, and its metabolic underpinnings. Keep those minds inspired, hearts light, and tails wagging,